The topic for today's session is physiology of bacteria. Physiology is a branch of science that deals with the normal functioning of microorganisms or their parts. Bacteria are microorganisms that are too small to be seen with the naked eye. Though unicellular, these bacteria have the ability to carry on various biochemical properties that help them to grow and reproduce. Growth in microbiology is defined as increase in the number of microorganisms rather than the increase in the size of the microorganism, which is usually defined in the biological system. So in microbiology, growth is usually defined as increase in the number of microorganisms. And this bacteria, as we all know, divides by binary fission. Binary fission is a process in which a parent cell divides to produce two daughter cells. So here the nuclear division precedes the cell division. The microorganism, there is a transverse septum that separates the two daughter cells from the parent cell. That is an ingrowth of a transverse septum across the bacteria that helps them to divide into two daughter cells. And the growth of the bacteria is usually expressed in terms of generation time or population doubling time. It means that a single bacteria gives rise to two bacteria. It is called as doubling time. So the growth is by geometric progression. So I will define generation time as the time required for a bacteria to divide into two daughter cells. That is called as the generation time or a population doubling time under optimum condition, that is keeping all other condition constant. When the organism divides to produce two daughter cells, the time required for a parent cell to divide into two daughter cells is called as the generation time or the population doubling time. And the growth is usually in the form of geometric progression, that is there is a constant growth in the number of organisms in relation to the time. That is, increase of the growth of the organism is in geometric progression. So the growth of the organism, that is the generation time of the organism varies from one organism to the other organism, one species to the other species, one genera to the other generation. Some organism has a shorter generation time. For example, lactobacillus. For example, you have this E. coli, which takes about 20 to 30 minutes to divide into two. That is the shortest generation time. Whereas some organism takes about hours, some organism even takes about days as their generation time. For example, leprobacilli divides in 20 days. That is the generation time of leprobacilli is 20 days. The growth of the bacteria can be seen in two culture systems. One is a batch culture system, the other one is called as a continuous culture system. Batch culture system shows us the characteristic growth pattern of an organism. All the four phases of bacteria can be easily seen in a batch culture system. Batch culture system is also called as a closed culture system, where all the nutrients are usually placed along with the inoculated microorganism, and the growth of organism is seen in a closed system, which shows the four phases of the bacterial growth curve. And in the continuous culture system, we regulate the growth of the organism, where the organisms are regulated at the log phase so that we can get the important metabolites from the microorganism. And this continuous culture system is mostly used in the industrial and research purposes to get the metabolite from the organism. And the continuous culture system can be regulated by a two ways. One is called as a chemostat, the other one is called as the turbidostat. So chemostat is the one, it's a system where you can see the change in the chemical nature of the medium. So you can keep on adding the nutrients so that the organism grows and produce the important metabolites. Whereas the turbidostat is the one that helps us to detect the growth of the organism by the turbidity. So here you can regulate the growth of the organism in logarithmic phase so that we can get the important metabolites from the organism. Whereas batch culture only shows the growth pattern of the organism. Now the organism growing on solid media is different from the organism growing on the liquid media. Organism which grows on the solid media forms discrete colonies on the medium. That is, a single parent cell divides to give rise to colonies. So these are called as a clone of cells from a single parent cell. Whereas the organism growing on a liquid media are usually the one that forms the turbidity in the solution. So this turbidity can be measured with the help of spectrophotometry. So growth is different in solid media as well as in the liquid media. And the growth, as I told you, is by geometric progression, and you can count the number of the organism. The bacterial count can be done in a particular system where different ways. The two important ways that are selected to count the number of the organism is a total cell count, where you can count the cell number, and the other one is called as a viable count, which is nothing but the counting the cell mass together. That is, the whole organism which are present in a particular solution can be counted in a cell mass. Now coming to the total cell count. The total cell count is nothing but the number of organisms present in a particular system. So this can be usually done by direct microscopy, where the smear of the organism is made and the organisms are calculated under the microscope. Apart from that, you can also calculate the microorganism by special devices which are called as Coulter counter or Petrov-Hauser counting chamber, which is a specially designed slide that helps to count the number of the bacteria under the microscope. And you can also go for plating method, where serial dilutions of the organism or the initial inoculum is done and grown on a particular solid media and count the number of the colonies. Apart from that, the third method that is used is called as a membrane filter method. Here a specially designed membranes are used to filter the microorganisms, and the retained microorganisms are grown on a solid media, incubated for their growth, and then count the number of organisms. Again, in the total cell count, you can go for special chemical assays, that is nothing but the nitrogen component that are present in the organism. Nitrogen is an important component that forms the basis of the organism's cell wall or the nucleic acid. So you can go for calculating the amount of nitrogen that is present to know the number of microorganisms. So this is nothing but the total cell count of the organism. The other one is the viable count. Viable in microbiology is defined as the number of organisms or produced from the bacterial cell. Viable means living. So each living organism, when placed on a solid media, divides into a colony. So that is nothing but the viable count. So the viable count can be done with the help of serial dilution technique that was discovered by Joseph Lister, where a series of test tubes are inoculated with saline water, and then inoculum is diluted to an extent that each test tube contains some number of organisms, and when plated on a medium, divides and produces the colony. Next method to count the cell mass is turbidity. That is, when the microorganisms are grown on liquid media, they usually makes the solution turbid, and this turbidity can be studied with the help of a special device which is called as the spectrophotometer. Apart from that, the other method that is used is called as the cell constituents measurements, where you can measure the amount of protein present in a particular organism. Protein is an important component of any organism. So you can count the amount of protein present in the organism by different chemical assays, that is Folin-Lovry method. You can go for Biuret method. So these are the methods that are used to count the number of bacteria present in a particular volume. Now coming to the other important topic here is the bacterial growth curve. As already discussed, bacteria usually divide by geometric progression, and a growth curve is usually obtained when the number of organisms growing are plotted against the time. So a growth curve is formed that helps us to know the 4 phases of bacteria. One is called as the lag phase, the other one is called as the log phase or exponential phase, stationary phase or the death phase. Now this is the curve that usually represents the 4 phases of bacteria, which is plotted again the number of organisms and in relation to the time. So geometric progression means constant increase in the number of organisms in relation to the time. So the 4 phases are lag phase, exponential phase also called as log phase, you have a stationary phase, and then finally the death phase. Now coming to the lag phase. This is the first phase when the organisms are inoculated or introduced into a new environment or a media. That is the phase where the organism will try to adjust themselves to the environment, and here in this phase there is increase in the cell size but not in the cell number. So in the lag phase, the organism will try to adjust and they produce important components such as enzymes and other chemical metabolites that helps them to divide in the next stage, which is called as the log phase or the exponential phase. The lag phase varies from one organism to the other organism depending upon the size of inoculum that we are adding, depending upon the media that is present, depending upon the environmental factors. So the lag phase varies from one organism to the other organism. Take for example, if an old culture is added to the fresh media, it will take some time to adjust and then go into the log phase. Whereas vigorously young culture, if it is added, it takes less or a shortened period of time and enters into the exponential phase. So lag phase is a phase that helps to organism to adjust to the environment. And here you can see the graph starting a little above the zero. That means initially the number of organisms are present. It is not zero. It is there are certain number of organisms that have been added to the media. That's why the graph usually starts a little above from the zero, not from the zero. Next, coming to the other phase, which is called as the exponential phase or the log phase. I am saying here it is log phase because there is a logarithmic increase in the number of organisms. That is nothing but the geometric progression. Slight increase in the curve you can see over here in the log phase, where the organism divides continuously by geometric progression till it reaches a certain level where they enter into the stationary phase. So the exponential phase of organism varies from one organism to the other organism, depends upon the genetic component of a particular organism. So here the organism divides by geometric progression. That is one cell dividing into 2, 2 dividing into 4, 4 to 8 till they reach n number of their generation. And the third phase is nothing but the stationary phase. You can see a straight line after exponential phase. There is a stationary phase which is usually indicated by a straight line, which shows an equilibrium between the cells that are newly formed and the cells that are dying. This type of curve can only seen in the mass culture because all the nutrients will be and inoculum are usually added only at once. There is no change in the medium or organism that are being added later on. So everything is in a closed system. So at a particular time after the cell divides, the organism enters into the stationary phase, which indicates that there is an exhaustion of the nutrients and there are toxic components that are produced by the microorganism, which usually kills the microorganism. So there is an equilibrium at this particular stage where the number of cells that are produced are equal to the number of cells that are dying. Now next immediate phase after stationary phase is the death phase. Death phase is completely opposite to the exponential phase. So there is a geometric decrease in the number of organisms, whereas in exponential phase we can see a geometric increase in the number of organisms. In the death phase it is geometric decrease in the number of organisms. And the death phase is due to three important reasons. One is the nutritional exhaustion. There is no nutrition left for the organism to take up and to carry out the metabolic activities. And next is the accumulation of toxic products due to the metabolic products of this organism. And the third one is the autolytic enzymes that are produced by the microorganism. So these are the four phases that helps us to determine the phases of bacteria, that is lag phase, exponential phase, stationary phase and the death phase. So it is during the exponential phase that most of the metabolites are usually produced that are of human importance. So here in a continuous culture system, they manipulate the batch culture, they keep on adding the fresh culture so that the organisms are vigorously multiplying and producing the metabolites which are of human values. Apart from the exponential phase, stationary phase also, the organism produces certain anti-metabolites such as penicillin, etc. that is of human importance to treat various types of diseases. So this is about the bacterial growth curve. The next topic in microbial physiology or bacterial physiology is the bacterial nutritions. So what are the nutritions that are usually required for the bacteria to survive in a particular environment? So here the nutrients that are required can be classified as macroelements and the growth elements. So the nutrients are nothing but the substances for the biosynthesis and energy production. So biosynthesis is nothing but synthesizing various components elements inside the microorganism, and energy production is for carrying out the metabolic activities. Now here the bacteria are usually made up of 80% of water, and remaining 20% are the organic and inorganic molecules, that is the proteins, the nucleic acids, the cell wall components, mucopeptides, etc. So 80% of bacteria is made up of water. Water acts as an important vehicle for transporting or it acts as a vehicle to taking up the nutrients from the external environment and eliminating the toxic waste that are produced in the organism due to metabolic activities. So the most important component with which the bacteria is made up of is the water, and the remaining 20%, as I told you, is made up of macro elements and the micro elements and the growth factors. Water, as I told you, is an important vehicle for taking up the nutrients from the surrounding environment and eliminating the toxic materials or the metabolites produced during the metabolic activity of the organism to the external environment, and it is a basic component of a protoplasm. And the other components, that is the 20% with which the bacteria is made up of, is with the macro elements and the micro elements. So the macro elements are the one that helps up to build the important components of the organism, such as protein, nucleic acid, cell walls, etc. Whereas microelements helps them to carry out different metabolic activities in the environment. That is, example of microelements are zinc, manganese, molybdenum, copper, nickel, etc. And the growth factors are usually required by the microorganism in trace amount. That is, the example of the growth factors are vitamins, riboflavin, folic acid, etc. So these are an integral part of the microorganism to carry out the biochemical cycles in the environment. And here the organisms can be nutritionally classified depending upon the energy source and the carbon source. So energy source, you can classify the organism as phototrophs and chemotrophs. Phototrophs are the organism that requires chemical light energy for their synthesis of the food, whereas chemotrophs are the one that requires chemical energies to synthesize their food. Phototrophs helps to carry out different biogeochemical cycles in the environment, and they have the ability to use light as the source of energy, and they can utilize carbon dioxide and nitrogen to synthesize organic molecules. So depending upon the carbon source, the organism can be classified as autotrophs or heterotrophs. So depending upon the energy source, you have classified the organism as phototrophs and chemotrophs. One utilizes light energy, the other utilizes the chemical energy to synthesize their foods, whereas depending upon the carbon source, you can classify them as autotrophs or heterotrophs. So autotrophs, they can prepare their own food, whereas heterotrophs completely depend on preformed organic molecules that are formed from the autotrophs. So autotrophs can utilize carbon dioxide and nitrogen, whereas these heterotrophs do not have the ability to synthesize, and they completely depend upon their autotrophs for their source of nutrition. And these autotrophs are very important in the increasing the soil fertility of the soil, and heterotrophs can depend on autotroph for various components from simple sugar, that is glucose, to other majority of the components such as amino acid, nitrogen, etc. Now you can classify the organism depending upon the oxygen concentration required by the microorganism. Organism can be classified into two types. One is called as the aerobic organism, the other are called the anaerobic organism. Aerobic organism, the name itself says that it requires oxygen for their growth, whereas anaerobic organisms are the ones that do not require the oxygen for their growth. That is, they can survive only in the absence of oxygen. And the main reason behind the classifying this organism is presence or absence of an enzyme which is called as catalase enzyme. This enzyme in aerobic organism helps to degrade the hydrogen peroxide and other toxic compounds that are formed during oxidative phosphorylation, so that the organism can overcome the toxic effects produced by this hydrogen peroxide. Whereas the catalase enzyme is lacked in anaerobic organism. They do not have this catalase enzyme. Therefore, the presence of oxygen results in the formation of hydrogen peroxide that ultimately kills the organism. That's why they do not require any type of oxygen for their survival. And the molecular explanation about this classification is The aerobic organism have oxygen as a final electron acceptor, whereas anaerobic organism, as they don't have the oxygen, they take up hydrogen or other nitrates or other sulfate as final electron acceptor. And this aerobic organism derive their energy from oxidative phosphorylation, whereas anaerobic organism derive their energy from a shortcut of respiration which is called as fermentation, or they get it from substrate level phosphorylation. So oxidative phosphorylation, here they have a complete electron transport chain which helps to synthesize ATP from ADP and other components, whereas the anaerobic organism usually derive their energy from the substrate level phosphorylation, where the substrate itself acts as an electron donor and acts as an electron acceptor. So during this process of substrate level phosphorylation and fermentation, certain important metabolic products are produced by the organism that are of human values. Take for example fermentative products such as lactic acid, alcohol, etc. They are very important for the human, though they are not useful to the microorganism, but they are very useful to the human beings. And these organisms which are aerobic and anaerobic can be again classified depending upon to what level they can bear up the oxygen concentration. Obligate aerobes, obligate anaerobes, facultative aerobes and facultative anaerobes. Obligate aerobes are the organisms that are strict, that is in their oxygen requirement. They cannot survive in the absence of oxygen, whereas obligate anaerobes are the organisms that cannot survive in presence of oxygen. They are strictly anaerobic in nature, whereas facultative aerobes and facultative anaerobes are the ones that can survive in or absence of oxygen. They can manage themselves in or absence of oxygen. That's why they are called as facultative aerobes and facultative anaerobes, whereas obligate are strict in their oxygen requirement or absence of oxygen. The microorganism can also be classified depending upon the temperature. Temperature has a profound influence on the growth of microorganism, and these organism ranges from they have their own varying temperature. Every organism has its lower temperature, minimum optimum temperature and the maximum temperature. The temperature beyond the lowest, this organism will not be able to survive. That is called as bacteriostatic in nature. Whereas temperature increase beyond their maximum level, this organism gets killed due to the killing action of the temperature. That is called as bactericidal in nature. So the range of temperature varies from one organism to the other organism, one species to the other species, and one genera to the other genera. So the organism which grows at very low temperature, that is below 0 degree centigrade, are called as psychrophiles, whereas the organism surviving at optimum temperature are called as mesophiles, and the organism loving the highest temperature are called as the thermophiles. So psychrophiles, mesophiles and thermophiles. Psychrophiles growing at very low temperature, these are the organisms that usually causes the spoilage of the food in the refrigerators. And then you have mesophiles that are mostly the pathogenic group of microorganisms that causes diseases in human bodies. And then you have maximum temperature loving organism, that is thermophiles, that can withstand highest temperature beyond 100 to 200 degrees centigrade also. It is because of certain lipid components that are present in the organism that helps them to survive at a very high temperature. So the organisms based on temperature can either be psychrophile, it can be a mesophile, or it can be a thermophile. The other factor required for the growth of the microorganisms are moisture. That is, as already discussed, it contains 80% of water, and if the organism are subjected to drying, they usually are killed or they inhibit the enzyme activity of the organism. There are certain organisms which are highly sensitive to drying, for example Trypanosoma pallidum, which causes the disease called as syphilis. So moisture is a very important component for the microorganisms growth. Next is hydrogen ion concentration, that is nothing but the pH. There are certain organisms that can survive in acidic pH. There are certain organisms that can survive in normal pH, organisms surviving at alkaline pH. So based on that, they can be classified as acidophiles, Neutrophiles and basophiles. Acidophiles are acid loving. They can survive at a pH of 3 to 5, whereas neutrophiles can survive at a pH range of 7 to 7.5, whereas acidophiles can survive at a range more than 7, that is till 8 to 9. Mostly the Vibrio cholera are said to be basophilic in nature, whereas acidophiles usually survive on the acidic pH. Now the effect of light on the microorganism, light is for the phototrophic microorganism, light is a very essential component, but due to the exposure to the UV rays, there is a possibility that the organism might get killed. So the effect of light on the microorganism is due to the UV radiation that usually attacks the nucleic acid and kills the microorganism by attacking the thymine dimer, forming a thymine dimers in the DNA molecule, thereby inhibiting the replication and ultimately killing the microorganism. Now next important factor is the osmotic effect. Now when the organism is introduced into an environment having hypotonic or hypotonic solution, the organism when introduced into the hypotonic solution, the organism dies due to plasmolysis, whereas in the hypotonic solution it gets killed due to osmolysis. So the osmotic effect also has a profound effect on the microorganism. Next, coming to the pressure, the organism usually are subjected to pressure of 1 atmosphere, that is they can survive satisfactorily at that particular pressure. As the pressure goes on increasing, it can rupture the cell wall and kill the microorganism. But there are certain organisms which are called as barotolerant that can that can survive the increased pressure in a particular environment. Ultimately, coming to the mechanical and sonic stress, the organism get usually killed due to sonic stress by rupturing the cell wall of the organism. So these are the basic factors that makes up the microorganism and their environment where they usually survive in a particular environment.